So based on these results, we can conclude that Roundup transform and glyphosate presence in aquatic environments do cause DNA damage in the streaked brocolod, and we can assume based off this that it can also cause damage to other species. We can also see, however, that due to the differences in the way that the DNA damage was observed in the cells among different concentrations, it shows that the erythrocyte cells and gill cells do react differently to DNA damage. That is why it is important to examine other organs as well when conducting these experiments, because our organs do react differently. The explanation as to why there was different results among prolonged periods of time in the concentrations as noted in the gill cells treated with Roundup Transorb having only a significantly higher DNA damage at the 24-hour mark could be due to DNA repair systems or cell turnover, explaining why at the 96-hour mark there wasn't much of a difference compared to the 24-hour mark. Using this same theory, we can deduce that the highly observable differences in the concentrations as seen in the erythrocyte cells of the Roundup Transorb at the 24-hour and 96-hour marks could be due to the point that the DNA damage is too great for the repair enzymes to function properly. We also noted that there wasn't much of a difference. There was significantly DNA damage in the gill cells of the fish treated with glyphosate. However, it wasn't as extreme compared to the erythrocyte cells. One of the theories to explain this could be that the gill cells are more in direct contact with the actual herbicide present in the aquatic environment, so they become, I guess you could say, a little more resistant to DNA damage. However, we still need further studies to prove these points for the DNA repair and the gill resistance to the herbicides. We also noticed more significant DNA damage in Roundup Transorb than in the glyphosate itself. So one of the explanations is that the surfactants in the Roundup Transorb that keep the whole chemical compound together is what causes more of a DNA damage than just the active ingredient. So I think this was actually very important because we aren't applying glyphosate on its own. We're applying it in Roundup products, in Roundup Transorb products that are already created to function for a variety of different uses. So there's all these surfactants in these chemical products. And so it's important to note that possibly because of these surfactants as noted in this study, it could cause more DNA damage to different species, and I think that's just something that we need to keep focusing on and keep on evaluating, because it's inevitable that all these herbicides are going to be continued to be used in the future to treat all these different crops and not crop blend purposes as well. So I think this study is a good way of showing how a chemical with an active ingredient and the surfactants present can cause more damage than just the active ingredient itself. Thank you.